Holy cow, it's 7 o'clock and it's 2023. Thank you all for coming tonight. Next week, on January 11th, Carrie Schrader from the Department of Surgical Sciences will be here. That's in the School of Veterinary Medicine. And she's going to be talking about veterinary anesthesiology, One Health, and all species. So she does work not only with dogs and cats, but she also does a lot of work at the zoo and with some other animals that we don't think of quite so much as livestock and pets. So I'm looking forward to hearing what she has to say. On January 18th, one of the saddest topics we ever have to deal with, malting barley research. Oh, boo-hoo. Samples? Way to go.
Yeah. Library.
Yeah, library afterwards. Way to go. I was always really pleased when I first came here as an undergraduate researcher to see the Barley and Malt Lab. And this is the successor to the Barley and Malt Lab. And we're still doing really great research here. This is being done by the U.S. Department of Agriculture, Agriculture Research Service, Cereal Crops Research Unit. And Marcus Vinge will be here to talk about the work that he's doing on barley. Then on January 25th, I'll be here to talk about the Xenos paradox and the futures of science outreach. And that'll take up the month of January. How's that?
Perfect.
All right, I will stand back here. Oh, there's the X. Now I do the formal part. Way to go. Welcome everybody to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for the Division of Extension Wisconsin 4-H. And on behalf of those folks and our other co-organizers, PBS Wisconsin, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight, it's my pleasure to introduce to you Molly Zeller. She's lab manager at the DNA sequencing facility here at the Biotech Center. She's going to be speaking with us about DNA technologies upcoming for 2023. Molly, I get to ask you the 5 questions. The rules are, the answers don't have to be true, they just have to be believable. You can answer in any way that you would like. Molly, where were you born?
Madison.
Native?
Well, I didn't live here, but I was born here.
Oh, okay. Where'd you live?
Down by Platteville, Hazel Green.
How come I didn't know that?
I don't know.
So Hazel Green's right by Cuba City there. Okay.
Yes.
I went to UW Platteville, and there are things that happened in Cuba City and Hazel Green. And then where'd you go for high school?
Hazel Green, Southwestern.
Very good. And then where'd you go for undergrad?
UW Oshkosh.
And what did you study?
Cell and molecular biology.
Way to go. And do you have any advanced degrees?
I do. I have a master's in science, cell and molecular biology also, from Colorado State.
Oh, go Rams.
Yes.
I tell you. And this is a talk that you happen to have given?
I gave the talk previously in October, minus like one slide I had to add because new technologies are coming out.
Way to go. And this is in Cold Spring Harbor?
Yes, Cold Spring Harbor.
Good. You'll have to tell us about that because that's a pretty cool thing. Would you please join me in welcoming Molly Zeller to Wednesday Night at the Lab?
All right, well, I guess Tom did all the introducing for me, but I'm Molly, and just to kind of give you like an idea as to how things are going to go, I do have a talk, but for those lucky enough to be here, my lab is like kitty corner, and so we're going to actually go see these cool instruments. So it's going to be pretty neat. I'm already jazzed about that part. And on the way in, I did put some flow cells up at the top there. If you happen to walk in that door, you would have seen them. But don't worry, if you are here, I'll show you more later. But if you're walking out, you don't want to join us for the tour, take a look at the flow cells. So with that being said, yes, like Tom said, I did give this talk before at Cold Spring Harbor. I was invited to give a talk there for a programming in biology course, basically on DNA sequencing. They were learning Python, and I was there to teach them about DNA sequencing. So, all right, so just a kind of a quick overview of kind of how this talk is structured. First, I'm going to kind of tell you who I am, and why I'm actually talking to you about this today. A little bit of the history of sequencing, and then we're going to go into the technologies that are current and around in 2023. So we have Illumina, PacBio, Oxford Nanopore, and then there also are some newly launched technologies now that Illumina, some patents are expiring, and just a lot of new companies trying to get in the market. And we'll talk a little bit about some applications, not too much, maybe some library quality control and data quality control. And then if you are a QR code reader or you want to visit any of the links, I do have a lot of QR codes throughout here, so have your phone ready if that's something that you are interested in. Also, one more thing before I get into it. I will at the very end have an option for a QR code that will send an email directly to me, and I will send you this PDF back to you. So, technology, right? All right, so a little bit of an introduction. We are in the University of Wisconsin Biotechnology Center. I'm sure you all know this, but it's a really cool place. And we have a lot of cores, a lot of science happens in this building, and a lot of science actually serves the rest of the university. So it was founded in 1984, actually by the father of one of my colleagues, actually founded this. His name is Ron Neese. So we're located in Madison. Sorry, this is a talk that sometimes I'm not in Madison. So we have many cores. So we have DNA sequencing, which is the best core out of the group. We have our Gene Expression Center, which works with RNA. We have Bioinformatics Resource Center, genome editing in animal models, mass spec, and flow cytometry. So we have all of these cores that service a lot of the labs here on campus. So a lot of the labs can't buy big, expensive $1 million sequencers, so we have them run things on there and send the data back to the researchers here on campus. I have been working here for 6 years, and like Tom asked, I do have my master's in cell molecular biology. And if you can't tell by my enthusiasm, I really love my job, so it's a lot of fun. All right, so a little bit of history. We're going to go back to the 1970s, which is a time that I unfortunately have never been in, but there were two methods that were developed in the 1970s. They're called Maxim Gilbert and Sanger. And I know a lot of you probably have heard of Sanger sequencing. If you haven't, not a problem. I'm going to show you a little video in a second. But there's also Maxim Gilbert. And I'll be honest with you, when I first kind of entered the world, all I heard of was Sanger. I didn't know about Maxim Gilbert. But that was actually more popular at the time when sequencing was first introduced. And so Maxim Gilbert actually depends on the cleavage of specific sites at different nucleotide bonds. And it's kind of complicated, but there's radioactivity involved, there's a lot of labor involved. So I did provide a YouTube link here at the bottom, so if you want to, when you get the PDF from me later, you can go to that link. And then we have Sanger, which is actually, excuse me, sorry, is going to be another video that I'll show you, and it's actually really cool. They do kind of like the animated drawing to kind of show you how it happens, which is much more probably informative than me just explaining it back to you. And then this Maxim Gilbert diagram here, I did want to note that this method, they're only able to sequence 200 to 300 base pairs at one time for one read. Now that might not mean anything to you at this current moment, so just hold that piece of information in your mind for a couple of slides later when I start to blow your mind with what we can do now. So I am going to actually show you the video here. Here we go. Oh no, that's okay, we have this.
Oh hey, let's go back to the basics and explore the technology platform that has been regarded as the gold standard for many years. Yeah, you guessed it, I'm talking about Sanger sequencing by capillary electrophoresis. Many might ask, why is it called Sanger sequencing? Well, Sanger sequencing is named after the inventor of this groundbreaking technology, Dr. Frederick Sanger, who I like to call Fred, who developed this method over 40 years ago in the mid-70s. So what are the basics of Sanger sequencing? Let's take a look at our lab book. It all starts by having a short primer binding next to the region of interest. In the presence of the 4 nucleotides, the polymerase will extend the primer by adding on the complementary nucleotide from the template DNA strand. To find the exact composition of the DNA sequence, we need to bring this reaction to a defined stop, that allows us to identify the base at the very end of this particular DNA fragment. Sanger did this by removing an oxygen atom from the ribonucleotide. Such a nucleotide is called a dideoxide nucleotide. This is analogous of throwing a wrench into a gear. The polymerase enzyme can no longer add normal nucleotides into this DNA chain. The extension is stopped, and we now need to identify what it is. We identify the chain terminating nucleotide by a specific fluorescent dye, 4 specific colors to be exact. Sanger sequencing results in the formation of extension products of various lengths terminated with dideoxide nucleotides at the 3' end. The extension products are then separated by capillary electrophoresis, or CE. The molecules are injected by an electrical current into a long glass capillary filled with a gel polymer. And during CE, an electrical field is applied so that the negatively charged DNA fragments move toward the positive electrode. The speed at which the DNA fragment migrates through the medium is inversely proportional to its molecular weight. This process can separate the extension products by size at a resolution of 1 base. A laser then excites the dye-labeled DNA fragments as they pass through a tiny window at the end of the capillary. And the excited dye emits a light at a characteristic wavelength that is detected by a light sensor. Software can then interpret the detected signal and translate it into a base call. When the sequencing reaction is performed, in the presence of all 4 terminated nucleotides, you eventually get a pool of DNA fragments that are measured and separated base by base. What you will get in the end is a data file showing the sequence of the DNA in a colorful electropharogram, and a text file which you can use to answer the questions you may be asking. And that, in a nutshell, is Sanger sequencing.
So that is Sanger sequencing, and that is, honestly, it was probably it still is happening a lot today. However, we have decreased our use of Sanger sequencing because now a lot of the times now we can do a lot of things cheaper on a lot of the new technologies that I'll be talking about. And then also a fun fact, in 1980, Frederick Sanger and Walter Gilbert actually won the Nobel Prize for the developments on these technologies. No? Oh my goodness. Sorry. Okay. All right, so a little bit of history, or more history, I should say. These are just kind of a little bit of a timeline kind of showing you what genomes were actually sequenced and how kind of these events unfolded over time. So you can see in 1995, the first genome that was sequenced was Haemophilus influenza, and that was in 1995. Keep in mind, we only have Sanger sequencing right at that time point. Then we have yeast a couple years later, and then here, actually from UW-Madison, Fred Blattner sequenced E. coli K12, and that was either in this building or really close to this building. I'm not gonna lie, I forget. And then you can actually see, I bolded some key points out here for you. So in 2001, the first draft of the human genome was done. And then just shortly in 2003, the Human Genome Project completed to 99.99% accuracy the first full human genome. Now that was tremendous, and that was an extraordinary feat in and of itself. However, you can see a couple years down the road, 2022 was a really big year for genomics. We had the first gapless assembly for the human genome assembled. And this was done by the Telomere to Telomere Consortium. And the Telomere to Telomere Consortium is actually it's an open community-based effort that was formed to generate the first complete assembly of a human genome. And to kind of go off of that, this is the science paper actually came out, I think it was May of this year, came out and had this out. And you can actually see everything here in red was everything that the T2T consortium discovered. So everything that was in red was not found up until just 2022 when, or I guess it was probably 2021 when they did all the work. But that came out. So they did all that red stuff by using all of these new technologies. So Sanger did a lot of the work, yes, but using the new technologies, so long reads, sequencing reads that are kilobases long, was able to help figure that out. And then I do want to point out that they say the first gapless, asterisk, all chromosomes are now gapless except for the Y chromosome. So the Y chromosome, there's still a little bit of work that needs to be done, but I figured it's close enough, right? So. All right, so more history. And I kind of made two little graphs here, but we're going to look at the one on the left here first. And I put out two little points for you. First of all, and hopefully we can see this, but we have the 2001 is the first time point here. And you can see the line graph is all the way at the top. And if anybody can see that number, it is $100 million. That's how much it costs to sequence the first genome. Does anybody here have $100 million? If so, we need to be friends. So moving on, so then the first arrow I drew is pointing to 2006. Does anybody know kind of what maybe happened in 2006? I don't know if we have any science history buffs here. So Illumina actually came out with the genome analyzer then. So that's when Illumina kind of came out with one of their first instruments, and that was in 2006. And I will talk a little bit more about Illumina also down then. But the genome analyzer could generate 1 gigabase in a single run. So one day, 25 base pairs, 100 million reads. So do we all save that information from before where I said 200 to 300 base pairs in one read? Now we can do multiple reads. We have different strings, basically different strings of base pairs, but we're only doing 25 base pairs. Okay? So improvement, but also not quite an improvement. And I do want to say that at 2006, they actually weren't called Illumina, they were called Solexa at the time, and I don't know exactly when the merge happened. I think it was 2007. Yep, sorry, I do, 2007. All right, so that's the first kind of drop in pricing, right? Because we can do some cheaper sequencing for a little bit more, or with more data. The second arrow is 2010, 2011. Do you have any other knowledge of what might have happened then?
Next bio.
Sorry, what was it?
Next bio.
Next Bio.
Long read forums.
Okay, yeah, so yes, there are some long reads that come out around that time. So yes, we are going to get a little bit introduction into that around this time. And also, Illumina does come out with their HiSeq and the MiSeq, so a little bit more high throughput sequencing, which at that point we're going to do probably 300 base pairs and probably up to like 20 million reads, right? So we're getting there, all right? It's nothing yet, just let me tell you. I'm like 20 million reads, I'm like, that's a day's work right there. All right, so that's the second kind of drop in pricing. And then I kind of put that second graph there. Basically, all I did is I zoomed in from 2010 to 2011 to 2021. And you can see that there's less tapering off, right? There's less kind of dramatic drops in pricing. And I would say that that's mostly, due to one, there's a lot of technology out there, and it's hard to beat a lot of this new technology because it's out there and it's great. But I do want to point out that as we decrease these prices in sequencing, there are also a lot of things that increase in price or that add to the cost of sequencing, like data storage. So before, we could probably fit, a couple base pairs on like a flash drive, right? But now we're talking millions of reads. We're going to have to talk about like servers. You're going to need a lot of space to store all of this data. Fun fact, we have like a whole little room downstairs that's just full of servers to hold all of the data that we generate from our instruments. All right, so no more history for now, but we do have, I'm going to go into the technologies. And to kind of start with the technologies, and before we get into kind of like the companies and the machines that actually do all this, I want to point out we have two different kinds of sequencing. We have short read sequencing and long read sequencing. So short read sequencing is going to be basically anything that's kind of less than a KB, so less than a kilobase. And that's going to be done primarily on Illumina. And so the cost per base will be much lower than long read. It's going to work very well for SNP detection. So if we're looking for small sites, small nucleotide polymorphisms, we'll be able to look for that. They're great for gene expression because those tend to be smaller kind of fragments, and ChIP-seq. Those are the pros. The cons of short read sequencing is that we do have to do a lot of PCR. To generate all these fragments in order to sequence them, we have to do PCR, which can incorporate, errors, and we're not going to be sequencing that native DNA. So I'm going to mention native DNA a little bit later also, but native DNA is actually the DNA that comes out of the organism. So there's been no modifications, so no PCR done to it. we're not copying it in any way. We're taking that native DNA and we're putting it directly onto an instrument. That's going to be done on PacBio, on Oxford Nanopore. Illumina, however, we do a lot of PCR, and we're not actually sequencing that native DNA. And then we have, there's difficulty sequencing high GC regions, and that's just due to the chemistry. The machine has to be able to read these bases apart. And then there can be sometimes there can be difficulty in resolving substitutions, deletions, or duplications, especially if they're longer deletions. If we're only sequencing up to 300 base pairs on Illumina, if you have a deletion that's much longer than that, or an insertion that's much longer than that, it's going to be a little bit harder to map or to analyze. All right, so some pros and cons of long-read sequencing. So long-read sequencing, I put here that it's anything greater than 1 kb. And I'm saying that because the PacBio and ONT, we're actually going to be looking at 10 kb, 12 kb, 50, like we're talking much longer sequence. Like we're not, oh yeah, we have 1 kb. 1 kb is like the average size that I feel like less people kind of aim for. So Illumina, we go for less than 1 kb. Anything greater than 1 kb, we're going to want to try to sequence it on ONT or PacBio, or Oxford Nanopore or PacBio. So some pros for long-read sequencing: no PCR bias. So we're not going to be modifying that DNA. We're not going to be introducing any kind of potential PCR bias. So we can determine phasing, haplotypes, structural variations, and indels. And then we can also aid in de novo assembly by spanning low complexity and repetitive regions. So by looking at longer fragments of DNA, we can look at, we can actually see kind of the bigger picture. Imagine trying to put a puzzle together and, it's 1,000 piece and the puzzle's pieces are like, very, very small. It's hard to put together, right? Because you can't see that whole picture. But instead, let's say we have, a child's puzzle. There's like 5, 6 pieces, right? And you're trying to put them together. They're like this big, right? That's going to be much easier to put together because you can see more of the picture when you're trying to assemble it. We can determine naturally occurring base modifications, such as methylation. So when we're sequencing the native DNA, the DNA that actually comes out of the organism, it can be methylated. It can have different modifications to it. And if we're sequencing that, that will change the, let's say the electrical current as it moves through that sequencer. It'll change, and we can actually read that and then determine if those bases are methylated or modified in any way. That cannot happen on Illumina. Some cons: the cost per base is much higher. PacBio and ONT have not reached the same kind of throughput or cost effectiveness as Illumina has. They've got 5 or 6 years on them in the game. Throughput is low. We need more input DNA, and analysis tools are still evolving. So since Illumina's been around a lot longer, we have a lot more tools that can analyze all of the Illumina data. PacBio and ONT, that's still more of an evolution happening at that moment. And then, as I mentioned before, storage and analysis needs are greater, so we're generating a lot of data. We're generating terabytes of data at a time. You need to be able to store it if you want to sequence with long reads. So, forewarning, the next few slides is like my jam I get really excited about, so bear with me if I start to speed. So, these are our toys, all right? I have these, they're literally like feet away from us right now, okay? We will go see them. So for Illumina, we have our NovaSeq, which is the taller white one there, and then we actually have 4 of the smaller MySeqs right there. Fun fact, all of our Illumina instruments are named after female aviators. So we have Willa, we had an Amelia, she has now retired. We had a Sally. We have a lot of cool things, and I can explain more about that when we go see them. PacBio, we actually have 2 of those instruments. Those are called Sequel 2s. Their names are Peanut and Butter. So for PacBio, combine and make peanut butter. And then we have Oxford Nanopore. So the top one that looks like a square on the top is our GridION. That can sequence 5 flow cells at a time. And our Promethean is a 24-spot Promethean, which can generate also a lot of data. And we're going to now go into kind of how, what the differences are in each of these technologies. All right, so Illumina. So like I mentioned before, formerly called 2007. Illumina was actually founded in 1998. However, before it got into the sequencing game, it was actually just a microarray. All they did was microarrays. But in 2007, they acquired Solexa, and then that was just after Solexa released the genome analyzer. We can see that they are a very large company, over 9,000 employees. Their revenue is $4.5 billion. They are in San Diego, and the CEO is currently Francis D'Souza. I actually went out to San Diego in September. They announced their new instrument, which I will be showing you in a second here. In September, they announced their new instrument. They had like this Illumina Genomics Forum, and they had Obama was there to speak. They had Bill Gates there to speak, and they announced this new instrument. It was a really fascinating time. So 2022 was a very big year for genomics, and honestly, I'm really excited to see what 2023 is going to bring, if 2002 was any indication. I do have another video, and this is just kind of show you kind of how Illumina sequencing works and how it kind of compares and how it differs from Sanger. I do have to only show part of it though, because it's a very long video, so bear with me for just...
Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away.
So, that, I just kind of, sorry. Oh my goodness. Okay, so that was kind of, I just want to showcase kind of how that happens in comparison to Sanger. So Sanger, we had a whole bunch of kind of little fragments we had every single time, and those fragments individually moved through some gel. Here, what we're doing is we're actually reading the bases as they're being added to that strand. So every time that fluorescence and that image that they showed of all the cool, pretty lights, it's exactly what the instrument's reading, is those little lights lighting up every single time something is added. To kind of just give you an idea as to what kind of happens during the process. So what we get is we get genomic DNA, the DNA that we extract from the organism. And then what we do is we have to break it up into small little pieces. Because remember, short read sequencing, all we can do is less than a KB. And genomic DNA is megabases long, right? It's just this big jumble of DNA that's in every one of your cells, right? And so we break it up into small little fragments, we put these little adapters on it, and then we actually amplify it, and then we were able to put it onto that instrument. And then it does all that, does more amplification once it gets onto the instrument.
I have a question.
Yeah.
When you break it up,
Mm-hmm.
at some point, do they bring it together so you know what order they are in the original genomic sequence?
Yes. So okay. So the question was, once you break it up, does it get put back together in the end? Yes. And so that's actually kind of comes downstream. So once these fragments, they're small fragments, let's say 200 base pairs, right? They get put onto the instrument. Once it comes off the instrument, all those 200 little fragments then, by somebody much smarter than me, a bioinformaticist, I don't do data analysis. So once it comes off the machine, that's somebody else's job. But what happens is then they can take those fragments and just kind of map them. So it'll start to look like a tiling. They'll take like this 200 base pair fragment, if it overlaps with that, they can tile it and keep doing that with all those little fragments. So yes, it happens, but now you can kind of see, like, with 200 little base pairs, that's like trying to do one of those puzzles that's like really small and you've got 1,000 of them, right? But let's say you have kilobase long fragments, and you're like, okay, this is a much easier puzzle to put together. Does that kind of answer your question?
But I mean, in order to hold them together, there's a lot of overlap that has to happen.
Yes.
That's sufficiently enough so that you can match them.
Correct. Yeah, a lot of overlap does have to happen. And that's where you kind of get into the yield that comes off of these flow cells, right? So we're looking at yield, we're looking at millions of reads that you're going to need. And for a human genome that's 3.2 gigabases large, that's a lot of DNA, or that's a lot of genome to cover with a 200 base pair fragment. So you're going to need millions and millions of reads in order to put all these pieces together to have that overlap. All right, so here is now kind of where I'm going to get into a little bit of, we're going to go back to that first piece of information I told you about one read, right? So the aluminum iSeq currently has the longest, well, I do stand corrected because it did have the longest reads at 300 base pairs for the Illumina's instruments. But now Illumina did release a kit they can sequence on the next Seq 2000 that can also get up to 300 base pairs. So I guess my slide is outdated. Technology, you guys, it goes so fast. So the aluminum iSeq uses 4-channel chemistry, and that's going to generate maybe 20 million reads max. Okay, 20 million, like I said, it's all right. Then we have the Illumina NovaSeq 6000, and that's going to generate up to 10 billion reads per flow cell. Not million, billion. And I actually have one of those flow cells up at the top there. I have the S4 flow cell will generate 10 billion reads per flow cell. And that's going to use 2-channel chemistry. And before I get into the chemistries, I just want to point out, at the meeting that I went to in San Diego, the Illumina did announce their new instrument, which is called the NovaSeq X and the NovaSeq X Plus. That is going to be able to generate up to 25 billion reads per flow cell on their largest flow cell. So a lot of things happening. And we are going to be getting a NovaSeq X Plus. So fun fact, we are getting a very expensive instrument. So I mentioned 4-channel chemistry and 2-channel chemistry. So Illumina's chemistry is called sequencing by synthesis. I think the narrator in the video mentioned that. Basically, 4-channel chemistry is going to use a mixture of nucleotides that are labeled with 4 different fluorescent dyes. So basically, if you get the pink, we know that it's A. You get the blue, we know that it's G, and so on. However, that creates a lot of time because you have to read each of those colors separately. The camera can't read all 4 colors at once. It actually reads all the pink at once, all the green at once, and so on. So what they did is they now have 2-channel chemistry. So basically, they have the A is labeled with both red and green. G doesn't have any dye associated, so it's basically a dark base. And T is green, C is pink or red. And so basically in their imaging, they take 2 images. In their imaging, if they see both green and red in both images of that same spot, they know that it's A. If there's no color whatsoever, they know that it's G. only green is T, only red is C. However, you can kind of see, like, what if we have a long stretch of Gs? What is that flow cell going to look like? It's going to be dark, right? So we can kind of run into a lot of problems when there's a big stretch of Gs in the genome. But it's been working well so far, but you can see, though, we do take a little bit of caution when we do get a lot of Gs present in the data. And then one-channel chemistry, they actually incorporate a chemistry step in between the two imaging. So, for example, A, if that chemistry step, if the signal is cleaved and we don't see it in the second imaging step, then we know that's A. G, once again, is dark. I don't know what they have against G, but there it is. T, if the image stays on, and then C is kind of basically the opposite of A. So that's a little bit interesting, but it also makes things actually a lot faster. So one-channel and two-channel chemistry is much faster, which decreases basically the production time that we have in order to get data back out to customers. Okay, so I hope this video works because this video won't actually come out of. So I mentioned I was in San Diego. I got to actually see their new instrument. So the NovaSeq X Plus is right here. The video's not the greatest because it was me being like, oh my God, what is this? It's amazing. So just kind of show you, this is their new instrument that is actually going to be starting shipping. I think they're shipping out starting in the spring to all the places. So yeah, the flow cell that you see there, that will generate up to 25 billion reads off of that one flow cell. So I think minute tons. Okay. And so basically the Illumina Genomics forum that I went to, like I said, they had Obama, they had Bill Gates, like there was a lot of big names there, was a lot of people. And then everybody's really excited about what they call the genomics era. And while you can already see from my talk, I've talked about like the technology is just outstanding and it's crazy, it's amazing. But they actually announced that we can now have a $200 genome. Do you remember how much the first genome was? 100 million, right? So 100 million, and now we're down to $200. I can afford $200. So it's really fascinating. And then this is kind of a little chart I put on the side discussing or showcasing the NextSeq. We don't have the NextSeq, the NovaSeq that we do have, and then the NextSeq X, and just kind of showcasing what applications can be done. All right. Okay. So I do have another video. I have two more videos, but I just really like, because like they're much better at animations than I can in my words, explain kind of how this happens. So I make this little joke, I guess, but like being a lab manager, at a core facility, it's kind of like being a parent. I'm not a parent in real life, and I know you're not supposed to have favorites, right? But I do. And I'm sure, I don't know if other parents do, but I definitely have a favorite, and PacBio is my favorite. I was kind of introduced to it. I worked a lot with this when I wasn't the lab manager. I did a lot of the preps for this. And so it just kind of became one of my favorite technologies to work with. So we're gonna watch this video. I promise this one's like 2 minutes.
Introducing the PacBio Sequel II system, powered by single-molecule, real-time, or smart sequencing technology. Here's how smart sequencing works. First, from any sample type, ranging from viruses to vertebrates, DNA or RNA is isolated. Next, a SMRTbell library is created by ligating adapters to double-stranded DNA, creating a circular template. Primer and polymerase are added to the library that is placed on the instrument for sequencing. At the core of smart sequencing is the SMRT cell, which contains millions of tiny wells called zero-mode waveguides, or ZMWs. A single molecule of DNA is immobilized in the ZMWs, and as the polymerase incorporates labeled nucleotides, light is emitted. With this approach, nucleotide incorporation is measured in real time. With the Sequel II system, you can optimize your results with 2 sequencing modes. Use circular consensus sequencing mode to produce highly accurate long reads, known as hi-fi reads. Or use the continuous long read sequencing mode to generate the longest possible reads.
All right, so that is PacBio, and they did actually do a rebranding. I want to say it was either 2021 or 2022. So you can kind of see the logo is a little bit different, and that was actually. This is where I had to update my slides because I gave a talk on October 16th, and then October 24th or 25th, they announced a new instrument. So I was like completely outdated in like 2 weeks after I gave this talk. So as mentioned in the talk, you can kind of see basically what they took is they just took, remember, native DNA. DNA comes straight out of the organism. All they did is add on these little adapters, which is basically just like putting your shoes on in the morning to go walk outside. Not changing anything else, you're just putting shoes on to go do a job. These here is they're just putting on the little adapters in order to sequence, and then they just sequence that native DNA over and over and over again. And by doing that over and over and over again, that eliminates doesn't eliminate errors, but it reduces a lot of the errors that are present in the data, or like, any polymerase errors that should arise. And then kind of taking a step back and going back to history, and I know it was mentioned around the 2010, 2011 mark back in my timeline, they were founded in 2004, and they were originally named Nanofluidics. I don't know exactly when they changed their name to Pacific Biosciences, I should say. But in 2010, 2011, they came out with their first instrument, which was called the RS. And that was out with kind of it's a really big instrument. I liken it to, let's see, I always think of like a bus. It's not the size of a bus, but like it looks like it was massive, right? It's huge. The RS2, also very huge. That came out in 2013, and then in 2015 came the sequel. We actually acquired a sequel in 2017, and then since then we've just been going crazy with our PacBio services, and we've been doing very well and sequencing a lot. In 2019, they came out with the sequel 2. In 2020, they came out with the sequel 2E. So you can kind of see like this, they generate instruments fast, right? And so we have to think about how much do those instruments cost.
I'm Aaron Wenger. I'm a principal scientist at PacBio. Today I'm going to discuss how the high accuracy of PacBio sequencing sets it apart from other long-read sequencing.
Oh goodness gracious, guys.
The SQL2E system. It was released in the last quarter of 2020.
How do I do
It's
it?
proven to be an extremely
Hold on,
reliable
I got an
platform
idea.
with very low run failure rates.
Okay.
Provides scale such that most projects can be achieved in a single run.
Oh, that was scary. Okay. Very sorry, guys. My videos did work in line last time, I promise. Okay. So they create a lot of instruments. And so you see the last one was created, I didn't update this slide, I forgot. But the sequel 2E was in 2020. And then the Revio is the name of their new instrument. And I have a slide on it. I'll show you a picture of it in just a second. That was in 2022. So like every, 2 years, they're coming out with an instrument. That's a lot of fast science, which is amazing. All right, so then we have kind of a little comparison between the sequel, so that was their first instrument, and then the sequel 2, which is on the left here. And just to kind of put out, she mentioned in the video, they have ZMWs. And so the sequel 1, you can see it says Smart Cell 1M. That meant that they had 1 million wells. So they're going to generate 1 million reads, essentially, off of a smart cell. The sequel 2 can generate, or had 8 million ZMWs, or little wells on the chip, and they could basically generate 8 million reads, right? 1 million, 8 million, you're like, well, Illumina could do 10 billion, right? Yes, but they're also doing much smaller reads. This is going to be much more accurate and much longer. We're talking 10 to 12 kb compared to 200 base pairs, so 10,000, 12,000, much larger fragments. And then just kind of a little bit of a comparison about the runtime, so 20 hours and 30 hours. So, this is my addition. I showed you the last video, which was their announcement of the sequel 2. The QR code will actually take you to their announcement video for the Revio. I didn't want to show it here because it shows some really funky music, and I didn't want to start dancing in front of all of you, so I will leave that there for you guys to watch on your own. Dance at home. But that is their new instrument. It's called the Revio. It was just announced. They're going to start shipping, I want to say not next year, it is 2023. They're going to start shipping, I think, mid this year. So just to kind of give you an idea, another little comparison. So remember, the sequel 2 had 8 million ZMWs, right? So 8 million reads. The Revio is going to have 25 million ZMWs on their chip. And then we're actually going to shorten the runtime to about 24 hours instead of 30 hours. And then the independent stages is saying that instead of loading one time, I can load 4 separate times to kind of keep the data generating, right? I don't have to load 1 and wait. I can do 4 at a time. All right, so that's PacBio. That's my baby. So now I'm going to go on to the next technology. One more video, I swear. Promise. And this is going to be Oxford Nanopore. So Oxford Nanopore is based out of the UK, and they actually sequence in a very different manner than, say, Illumina and PacBio. And it's very fascinating.
Have you considered going solar in Wisconsin, but you're concerned about the huge price tag attached?
I do have some of these to show you also. I don't know if anybody caught that, but they have actually sequenced in space. Ah! Oh my goodness. So, pros and cons to ONT. Sorry, I shortened it to ONT because Oxford Nanopore can kind of be a lot to say multiple times. So pros, there are no limitations in the length of the molecules that can be sequenced. So remember, way back when I was talking about 200 base pairs, then I was talking about PacBio, 10,000 base pairs, 12,000 base pairs. There's no limit, right? Because the DNA just goes through that pore that we saw in the video and it keeps going. If there's more DNA, the more DNA just keeps getting sequenced. Once again, we're sequencing native DNA. All we're doing is just adding that little motor protein and that little tether on and sending it through that pore. Capable of detecting DNA and RNA modifications. So one thing I haven't mentioned up until now is RNA. We've been sequencing DNA, but Oxford Nanopore, you can actually sequence native RNA as well, which is mind-blowing in and of itself. And then their two smaller flow cells, the flongle and the min-ion, are actually portable. And that's what you saw up when they sent it up to space. And then some cons. It does have difficulty sequencing homopolymer regions. The error rate is higher than other platforms, platforms being Illumina, PacBio, and then you can't sequence the same strand multiple times. So like PacBio, where we just keep sequencing it over and over to kind of basically check for all those errors and reduce them. ONT, you can't. Once it goes through the pore, that's it. It's not going to suck it back up and go through again. Right. And then we do have just a quick note, the flow cells, the mid-ion, you can do one at a time. That was the little kind of square guy that's portable. The grid-ion, which we have up at the top here, that has 5, and the prometheon can do 24 at a time. And then just kind of give you an idea of yield, we can generate 15 to 30 gigs on mid-ion and the grid-ion. The prometheon can do 100 to 180 gigs of per flow cell. And then these are just a couple of images showing kind of where ONT has been sequenced. They've sequenced microbial DNA in Antarctica. We've got sequencing bacteria in space, Ebola in Guinea, and Zika virus in Brazil. So this is a lot, like, you can do a lot of field work with this, right? That's what makes ONT so fascinating, is because you can just take it and you can go, you can plug that into your computer. And then we also I want to point out the kind of up and coming technologies, right? So there are technologies that are coming out. Keep in mind, these are all more short read sequencers. So we have Ultima Genomics is coming out. Their instrument, I think, is called the UG100. The Singular Genomics is called the G4, I think. And the Element Biosciences, that's called the Aviti. So these are all instruments that are kind of competing with Illumina. They're coming out with short read sequencing. And I want to point out that Ultima Genomics doesn't operate on your typical kind of flow cell where there's some glass put together and you just put liquid through it. They actually are using what's called a spinning wafer. And I don't know how, this photo here, okay? This is the photo that they have on their website. I don't think it's like super accurate, but like basically what they do is like the liquid is just spread across this wafer as it spins, and that's how the sequencing happens. So very different. I thought it was very fascinating and different compared to the rest of the flow cells that we've seen. So in the interest of time, I am going to kind of a couple of the next slides, just kind of give you an idea as to what can go on these instruments. So we have DNA sequencing. So DNA sequencing is a very vague topic, right? We're going to sequence DNA, but what are you doing with that DNA? What are you doing with that data? Are you doing whole genome sequencing? That's where we're going to fragment all those pieces, and then we're going to basically put them all together, and that's just all of the DNA. We're going to sequence all of the DNA present in that sample. Exome sequencing, however, or I should say targeted, you can also do this the same way, but you're going to take all those, fragment them, but you're then going to take little probes and kind of pull out the regions that you are interested in, or the regions for, say, that exome, or so basically you only are going to sequence just the pieces that you're interested in, or the exome, which is the protein coding regions. This is actually something that I worked on last May. I presented on this. PacBio is actually coming out with a targeted application. Not coming out, they have come out with it. And this is something I did. Basically, I took human samples, and up at the top, I hope it's big enough to see, but I targeted 4 genes that I wanted to sequence. And you can see their lengths up there. So their lengths are in base pairs. The genes are, 230,000 base pairs long up to 1 megabase long, right? So I'm looking at genes that are very long, and we're trying to look at just those genes specifically rather than looking at the whole genome and pulling those genes out. And then we have other applications like the metagenome. So if you want to sequence not just, say, like the human sample, right? We're also going to sequence, fecal samples, saliva samples, anything that's got a community of organisms in it. We want to sequence all of that DNA that's within that sample. And if you want to know, yes, we do extract DNA from poop here. So blood, saliva, but fecal samples are very fun. I'm going to skip this one. And then I did mention RNA briefly. I don't do RNA. That's what the Gene Expression Center here is for in the Biotechnology Center. But if you wanted to, I made another QR code that will kind of explain. It's like a 100-page document. I don't expect you to read all of them. But if you want an idea as kind of what different types of RNA sequencing are out there, that QR code does take you directly to it. All right. And then going into just a quick note on extraction. This is actually DNA being extracted from a sample. I don't know what sample, but basically we just basically add in a bunch of like lysis buffers, open up the cells, the DNA comes out, kind of globs onto this little, you can kind of see a little disc in that far image there. But that's DNA just hanging off of that disc. And then we pull that out and we clean it a little bit. And that's what we use to put onto PacBio or ONT. That is the DNA that's going on and being sequenced. So it's very, very fascinating. So with that, I'm going to skip over the last couple of slides because those were part of my talk at Cold Spring. But I'm going to go to this slide, which is my thank you slide. And if you would like me to send the presentation to you, I'd be happy to. Scan the QR code. It will literally just pull up your email, and it will send me an email. And I will send you one back. And I will say hello. So with that, I just want to say thank you. This is fascinating, and I hope you guys now have a good appreciation for DNA sequencing, and I'd be happy to take anybody on a tour that wants to go see the cool instruments. So.
Any questions? All right.
Hi. So I used to run a sequencing lab in a previous generation, but so we only need a little bit by PCR, but for the PacBio, what's the amount of DNA that you need?
That is a great point. So what is the amount of DNA? So yeah, so for Illumina, we really only need, we can generate off of like a nanogram of DNA, okay? Very small amount. For PacBio and probably for Oxford Nanopore, we're talking micrograms of DNA. There are applications where you can use less, but it's a little bit more finagling. But if you want a really good, robust data and robust experience, we're talking probably 5 micrograms of DNA.
And how do you do single cell DNA sequencing if it's that small?
We don't.
Okay.
We do have single cell RNA sequencing here, but that's also done by the Gene Expression Center. So.
Okay.
Yeah.
RNA, DNA, sometimes I can't tell them apart.
I know, but DNA is cooler.
What? What was that?
Nothing?
Okay, good, thank you. Other questions?
Early on, you used indel, insert,
Yes.
delete?
Yeah.
Okay.
Yep, so yeah, indel would just be like an insertion or deletion. Sorry, yeah, sometimes they'll slip through, so.
Just wanted to make sure I didn't interpret it.
Yep.
So 5 years ago, when I was doing shticks here and showing people around, I'd talk about the Holy Grail being all your DNA in one day for 1K. In other words, can you get all the human DNA, a patient's DNA sequenced in a day for about $1,000, based on the idea that an MRI of your knee might cost about $1,000, and you get only one image, but here you get your whole book. You said $200?
$200 in one day.
In a day.
24 hours, yes.
So while for clinically stuff, you could say at the hospital, here's a sample. That's, I'm
correct.
talking
So-
Holy Grail here.
All right,
but
so
yeah.
I'll clarify. So one day for the sequencing run to happen, a little bit of upfront prep, maybe a day, not even a day,
Okay.
8 hours of work. Sequencing run, and then the data analysis, however long that takes, so.
And the first 4 letters of analysis are, so it takes a long time. How long
Yes.
how long is that?
I'm not sure. I want to say probably not that long, like another
Okay.
day.
No, I don't mean
Something
to, I'm just fascinated that, oh my goodness, the potential for really rapid clinical, useful
It's,
stuff is getting there pretty fast.
it really, truly is.
Okay,
You have
wow.
more questions up there?
Oh, you're telling me to go back there?
Yeah.
Okay, good. Whose hands was it? Oh, we're back here. Sorry about that.
So this is partly my field, and I used to work with the Human Variome Project, which was run out of Australia. The problem is now not sequencing, as you very elegantly showed us today. It's interpretation.
Yes.
And we don't know how to do interpretation of many of the variants that we find.
Correct. Yeah. It's analysis. It's storage. It's basically everything that comes. Like, we've got it figured out. We know how to do this fast and cheap, but it's getting that data analyzed and annotated and published and also just kind of organized. Like, there are a lot of the consortiums out there that are also just trying to organize it, because you have labs here on campus trying to do something. You have labs in Chicago trying to do something. Like, we also have to organize this and try to get this to be, we need standards also. So there's so many more cool things yet to come and a lot of advancements being made. But yeah, analysis is also one of them. And I wish I could speak more on the analysis, but I got to worry about this. I can't keep up with all that too. So.
This is so cool. I'm so impressed. And you gave a great talk just to kind of give an overview. Are there safety considerations when an investigator submits samples, like fecal samples? Or I'm thinking of Tony Goldberg, who has he has his own staff at the Kabali Eco Health facility in Africa, and they're collecting all kinds of fecal, urine samples. Are there safety considerations for the analyst here when they get those?
So when the samples come in truly as fecal samples, yes, there are concerns, and we have a BSL-2 space upstairs. And when the samples come in, we do treat everything under our BSL-2 and our biosafety protocol. So we do treat everything as it's going to be harmful. But once that DNA is extracted, there's no concern for that. So basically, once it's no longer within that cell or that community of cells, I should say, and it's just straight DNA.
So there's nothing that would require a BSL-3.
Correct.
Everything is within a BSL-2?
Not here, not for us. We just have up to BSL-2.
Okay.
So.
Can you mention what BSL-2 and 3 are?
Sorry. So BSL-2 is biosafety level. So everything basically in a lab is BSL-1, which is lab coat, goggles, gloves. BSL-2, you're going to want to have a little bit more coverings on. BSL-3, then there's BSL-4 also, which it's just very dangerous things. Like, I don't know, what's a really dangerous organism? I can't even think right now. Ebola, Zika, yeah, I just mentioned them. But yeah, so we only deal up to BSL-2, and we do actually handle what are now considered human pandemic samples. So COVID has actually thrown a big wrench in our plan. So anytime a human sample comes in now, we have to treat it as human pandemic if it's been collected after November of 2019. So little extra precautions we got to take, but we have done them safely.
Given this is generally a random process, you have errors. You mentioned a lot of errors. I suppose that there's a mechanism for correcting errors or detecting errors within this. Let's say you run through a sample and you run it through multiple times, or not the same sample, but basically you're making comparisons between the results of one pass versus another pass to ensure and make a decision about which
how accurate
this is, and so how does that operate and what's going on there? Is that part of the analysis, or is that part of what the machine does?
Machine. So I will show you a slide that I did not actually show you. If we go, nope, I didn't. Sorry, it's this one. So this here, these little graphs I'm actually showing you are Q30 scores, or they're Q30 graphs. So basically, the Q30 score is calculated by the instrument. So as it's sequencing, and you kind of see these are the cycles as we go that way, and then the percentage Q30. So obviously, high percentage means more accurate. So Q30, and you're probably like, what is Q30 even? It corresponds to the chance that a base call is wrong. So it's not easy to describe. And I've probably spent quite a lot of my time trying to figure out how to explain it and how to get it to be best. Basically, if I show here, so it's based on the Fred score. So the Fred score quality, 10 means that 1 in 10 chances that it is incorrect. So that is the Q, that would be like a Q10. So Q30 score is what Illumina basically bases all of their calls on. And basically the higher percentage means the better the call is. And Q30 means 1 in 1,000, so 99.9% chance, or percent, that call is right. And so it's basically, I don't exactly know how the math is done, but that is how we determine and how Illumina has basically, how it comes off when it comes off our machines. We look at that and we're like, okay, Illumina thinks this is a good run, this is the Q30 scores, the data quality is great. Does that kind of answer your question?
Yeah, I mean, I could want to know how it's making that distinction underneath with other sequencing of multiple.
I would also like to know.
Yeah.
It's more math, and I'd like to.
Little computers doing it for us.
Computers, yes. Technology does it.
Is there something roughly the equivalent of the Library of Congress where all of this data gets put for the use of other researchers, or are people hanging on to their proprietary stuff and not sharing?
I'm not going to say officially both, but probably both. I know people are going to sequence, and labs are going to do it, but when it comes to publishing their work, they do have to report the genome to, there is a database. I'm blanking on the database's name, but when it does get published into a reputable journal, that data does get stored and information on the genome. And if you go to NCBI's website, you can actually look up kind of the genomes and then what kind of, how they were done and the methods, and so.
NCBI is?
Sorry, yeah. NCBI is the National Center Biotechnology Information, yeah, okay.
Okay.
I've said it so many times I forget what the letters are.
Yeah. Thank you.
There was a recent retraction of a paper by looking at their DNA evidence that they cut and pasted it. And I wonder, with the new technology you're talking about, if people will detect people messing with their data collection.
I do think that. I think now with a lot of these error corrections and a lot of the technologies now we're coming out with, I think a lot of things are going to come out of the woodworks, not necessarily in a bad way. Like that is an unfortunate example of somebody cutting and pasting, but I do think a lot of corrections or a lot of the genomes that have been published, if we're coming back and let's say we go back to C. elegans that was sequenced in my timeline there, we can now go back and sequence any sort of different kinds of, if you have C. elegans from somebody's lab and C. elegans from here, what are their differences? And not to detract too much from your question, but it kind of brought me to another point of what's called the pan-genome, and that's actually, taking all of the genome. So like, I think we mentioned barley before, right? So we sequenced barley, but that was one individual out of how much barley is on, in the United States, how many things. So like the pan-genome is taking and putting all of those different genomes together and then kind of like, like I can't sequence my genome and say it's going to be the same as Tom's, right? So, minus the X and Y chromosomes, but, it's taking all that information and being able to compare everything across. So yes, I do think that, we are going to be able to correct a lot of things for good or for bad. But I also think it's going to be just beneficial in order to like get more of a community of genomes. That kind of went on a little tangent, but hopefully it's okay.
So your place is a business.
Yes.
You have to cover your costs.
Yes.
What kind of cost how much does it cost a researcher to use your services on a per base? It used to be $1.25 a base. Something like that.
Yeah. So
long ago.
So that's a great question. So it depends on where the customer starts. So let's say they just start and they have a blood sample, right? We do have a charge for us to extract, and that's going to be probably couple $100, right, for us to extract the DNA. Then we have to prep that sample. that's probably going to be looking at, if we're talking, it's probably going to be $500 to prep that sample, to put it on the instrument. If we're going to do Illumina sequencing, let's say that was a human sample, we're going to put it on our shared sequencing service on the NovaSeq, so the NovaSeq 6000, and we can sequence for $6.35 for 1 million reads. So for a human genome, if you want 30x coverage, you're probably looking at like, I'm just going to round up to 300 million reads. So 300 times $6.35, that's how much it'll cost for us to sequence your genome at 30x coverage. So less than, yeah, yes. Very cheap per base, yes.
I was a little worried to ask this question because it might show some pretty big ignorance on my part, but here it goes after listening to, is all of this just for, it's not going to tell you anything other than it's going to be used for comparison work? Like saying it's somebody, this is somebody like a crime person at a crime scene, this is somebody's DNA because it has this, this, and this. Not that that's necessarily telling you anything about the person, but it doesn't match this. And everything I hear doesn't, seems like it's telling you anything other than what you can compare with other DNA.
So, great question, and you're not, I wouldn't say it's not, it's kind of a mixture of both. So yes, do we use it to compare? Absolutely. We're using it to compare, like I said, the pan-genomes. We're using it, we're going to want to compare one barley genome to, this strain of barley, right? Strain. But, like we're looking at the comparisons. We want to see that. We want to see, but the de novo part of it, of doing, sequencing something that's, say, brand new that hasn't been done before, or you sequence that's a brand new discovery. Or let's say everything's all been done on Illumina up until now, and you sequence on on PacBio or ONT, and you're like, oh my goodness, you can actually see something in if more resolution. So that's a discovery in of itself, is getting that resolution of that genome. So rather than looking at something that was scrappily put together of just putting two little pieces and pieces and pieces together, now you have that whole picture, and the resolution is much larger or higher. So yes, absolutely yes, it does go towards comparisons, but it's also being, you're able to discover new genes and new things. And what I find also fascinating is, like, when I mentioned the metagenomics part of it, if we're looking at, let's take a fecal sample, right? And we've just been able to sequence, basically using 16S, we're pulling out, okay, we know this organism's in there, we know this organism is in there, but let's say we're able to, sequence at a much higher resolution of that DNA that's in that metagenomic sample, that fecal sample. We could discover new organisms that maybe are present. Maybe that's, in the clinical or the health field, somebody who's working with, like a digestive expert or something. So yes, there's new discoveries, but also comparisons. So does that answer, maybe? Sure. Okay.
Do you happen to know what Exact Sciences uses? Because they must have something similar in their labs to be able to do an analysis for cancer, for example. Do you know what instruments they use for their systems?
I don't know exactly. I want to say that I know that they do have a suite of Illumina instruments, but I don't know exactly what ones they have. So the Illumina instruments, they probably have a MySeq, probably NextSeqs, and I don't know if they have a NovaSeq, but they do use Illumina instruments. So that's about all I got for you on that one.
Oh, I guessed wrong on which side of the street to be.
It's good exercise.
Oh, is there bad exercise? I noticed that. Thank you.
Sorry.
With this cool technology, part of me also thinks about how accessible this will be years from now. So if a sequence has been determined on a nanopore instrument, or let's say PacBio, could it be recovered or looked at again years later when the technology gets presumably better? Part of me is thinking that because, I can no longer play my VHS tapes. Or my track.
Yeah. Yeah.
I'm just wondering if anybody's thinking about that to keep the data consistently accessible.
Yeah, so there's kind of a couple different ways to kind of think about that. One is storage of the actual DNA sample or that tissue sample. So let's say we have a sample and we extract DNA. We've sequenced it out Illumina. Well, let's say somebody comes back a couple years later and like, hey, we want to use that DNA, but we actually want to sequence it on PacBio. So we can, there's that aspect of it, but then there's also like, looking at, and I think I actually read an article, I want to say it came out today on Science, about how there's this tool coming out where you can actually put all sorts, like it's an analysis tool, and you can take Illumina data, PacBio data, whatever data, and it will take it no matter what format it's in and be able to analyze it all together. So there's a lot of aspects to your question about can we, come back to this. And one, yes, we have to store and preserve said samples. Not only store the samples, we have to store the libraries that we make. We have to store the data that we make. But once again, we're creating so much data. Where does it go? Like me, I'm like, well, it goes to the cloud, right? And the cloud's big and it can hold everything. It can't. And that storage costs money. So yeah, there's a lot of fascinating aspects to that question. I think about it a lot too. I'm like, also, if this is going to become more accessible, my job as a core facility that's a business, will everybody be able to just do it on their own? Well, do I have job security? So, a lot of things. Like there's a lot of facets to that.
The National Academy of Sciences has a paper out from 2019, talks about reproducibility and replication, and one of the pieces that they have concerns about on reproducibility is that computer programs change all the time, and this is basically a big computer, some sensors and a lot of computer. And every time there's an upgrade to the operating system or of the machine or a change in the code, there's an issue of reproducibility because there's new code, corrections to the code, etc. Do you have to, are you aware of having to upgrade the software on these machines on a continuous basis? Do they have upgrades on it? And then the other is, do you get requests to redo something because the software has changed?
Okay, good question. Do the instruments themselves? No. The instruments usually come with one kind of standard, like the software is locked in. When that instrument's released, that software is locked in. There might be kind of some minor updates, like from version 1 to version 1.1 with maybe some bugs or something, but it's not often that we have to update the instruments. So I would say no to that one, no. And then do we have customers wanting to redo? Not on the sequencing side of things. They usually, if it happens on the analysis side, I don't really know the answer to that, but not on the sequencing unless somebody wants more data. And then we usually give them more data. And yes, there is always the concern of, well, you ran it the first time on flow cell A, but this time you're running it on flow cell B. Was there any environmental changes? Was the temperature in that room different? Were the flow cells, maybe one was slightly, they're different lots, right? So yeah, there's also, the consideration of lots of reagents and lots of flow cells. Being able, nobody really asked to redo it. I think people just kind of take that information into consideration when analyzing things from two different places. Or if I have to give a customer more data, they're like, okay, well, you ran it on flow cell A this time and flow cell B this time. We'll take that information, but the data is, since that software doesn't change, it's basically kind of put together as one. Does that make sense? Maybe.
Well, I mean, it makes sense, but my own sense of being a computer programmer is everything changes all the time.
Yes.
And that there's always bugs.
Yes.
That might change the results of things.
Correct. Yeah. And
in many cases, the questions that the National Academy of Sciences raised were new versions of SAS or new versions of R. People that, and then those kind of results can make changes in the published results based upon new software.
Yeah, and that's why the Illumina doesn't change. Like our NovaSeq software, I think we had to upgrade it once, just to deal with a kind of a new kit format that they had coming out with. But we got that in 2018 and just upgraded it the once. So the software is set. When they want to upgrade it, they go to a new machine, right? So that's another $1 million out our window.
So Molly, you offered to give folks who would
Yes.
like a little tour before we head over to the library. So if this is a good time, why don't we thank Molly for a great talk, and she's going to lead you on a little tour.
Thank you.
Just an event because I'm completely ignorant. What is a read?
Read. It's basically just like that's like that strand of data. So one simple. So we have like they showed in Illumina those little fragments. It's basically just going down that one fragment there, but on that fragment or on that chip there were millions of other little fragments. That's what you put together.
Million reads,
Yes.
Right? And it does that all at once, parallel.
Yes. Yep. How do I exit?